Hello, everyone. Thank you very much for coming to today's National Chemistry Week event with AIAA Los Angeles Las Vegas section and also the SCALAS, the Southern California American Chemical Society. Today we have a fun talk, a very exciting and a very good speaker. Um, uh, Brenda, Miss Brenda Ip, completed her BS in biochemistry with a major in forensic science at the Cal State Los Angeles University in 2017. She is now pursuing her PhD in the biology for aging program with the University of Southern California, USC, and the Buck Institute of Research on Aging. She is working under Dr. John Newman, studying the roles of ketone bodies and the ketone esters as regulators in aging. So without further ado, let's welcome Brenda for the interesting talk today.
Thanks, Kenneth, for the introduction. I want to thank the organizers too for keeping National Chemistry Week alive, and Kenneth for putting this all together, and Dr. Kaluri for actually asking me to speak today. So thank you, Dr. Kaluri. Kenneth said that I am a PhD student in biology of aging. Our program is affiliated with the Buck Institute here. You can see here, this is a very majestic view of the Buck on a cloudy day. And today I'll be talking about or providing insights into ketone bodies in aging. But first, before I start, you might be wondering, where is the Buck Institute? So to give you some perspective, this is San Francisco here. And then across the bridge, we have East Bay, which is Oakland and Berkeley and Richmond. So if you cross either of these bridges and make your way up north, you make your way into Marin County. And Marin County is part of the North Bay area. And then the Buck Institute is on top of a hill, as you saw earlier, and it's in a quaint town called Novato. So if anyone's interested in ever visiting after COVID's done, after pandemic, just let me know, and I'd be very happy to give you a tour of the Buck Institute. Our mission here is that we have a goal to end the threat of age-related diseases for this and future generations. And who's ever interested in checking out their website, I've posted it here also. And then just to make my intro a little bit more complete, I actually studied aging in my undergrad career. I studied it in plants. So our group goal was to prolong the life of plants, and it had two implications. One was to prevent food storage. So if you think about it, when we eat vegetables, we eat the leaves of the plant and not really the seeds. So our goal was to try to prevent the plant from senescing and producing seeds, and kind of just prolonging its vegetative stage. So to make it produce endless leaf counts. And so that was one implication. The other implication was to study aging pathways aging conserved pathways across species, which brings me to my work now. I study mice and aging. And to give you some perspective, I study young, middle age, and older mice. So young would be like a 3 to 6 month old, and that's equivalent to 20 years of age in humans. Middle age is 12 to 14, equivalent to 40 years old in humans, and then older, 22 to 26. Some people even use older mice, but this is around 60 years old in humans. So back to my talk. So today I'll be providing insights into ketone bodies and aging. And so this is the flow. I'm going to introduce what ketone bodies are, the roles of ketone bodies in aging, and how I study ketone bodies in my research. So since it is National Chemistry Week, I figured I would extend my appreciation to ketone-containing compounds. So on Monday, we heard about amoxicillin, and I figured I would just throw that here. On Tuesday, we heard about Dr. Kaluri and good nutrition and healthy foods So I figured I would throw something out there, and I threw out this type of flavonoid here, which is found in plants, and we consume them for their antioxidant properties. So anthocyanin is, so it's for their antioxidant properties when we consume it. And our favorite solvent in the lab, which is acetone, which is also a ketone body. So what are ketone bodies? Well, ketone bodies consist of 3 molecules. It's converted from, so the first molecule is acetoacetate, which makes acetone, which we normally breathe out, but then it also produces, quote on this, I put a quote around here because it's not really, chemists would say this is not a ketone body, and they're right, it's not, but acetoacetate converts into beta-hydroxybutyrate. And for the rest of the talk, I just want you to think about beta-hydroxybutyrate as a package. So this important package that needs to be flown in the blood circulation to get where it needs to be. So those are the 3 ketone bodies. So ketone bodies, or productions of ketone bodies, has dated back to bacteria. It's a fun fact here that bacteria actually produce polymers of beta-hydroxybutyrate as energy storage. So when the bacteria senses a environment where nutrients is limited, they create this type of polymer for energy storage and fuel. And we actually take advantage of this biosynthesis, and we actually use it to make biopolymers as plastic substitute. So that's some sort of related fun fact for you. And then, so bacteria makes polymers of BHB, but in humans, we don't make polymers. We just make, the BHB molecule. And this happens especially in the brain when glucose levels are low. So glucose is our main primary fuel in very important organs like the brain, the heart, the kidney. But when glucose levels are low, we're able to make ketone bodies, and we can use it as alternative fuel source. So here, normally, we have glucose entering the blood-brain barrier via glucose transporters. But when glucose is scarce, we use beta-hydroxybutyrate, and also they also use monocarboxylate transporters, otherwise known as MCTs. And we heard a little bit about serum albumin and how that is a very sticky protein, and that actually helps the facilitation of drugs or even small molecules to get to their target tissues and do whatever they have to do. So here, once beta-hydroxybutyrate crosses the blood-brain barrier, the astrocytes actually shuttle them to neurons, so neurons can actually meet the energy demand, because neurons, they're very active in the brain. They're responsible for our neuroactivity and like firing off synapses and chemical neurotransmitters. So they're very important, and it's important to keep the demand, maintain the energy demand in the brain. So in humans, where are ketone bodies made and what triggers it? Well, ketone bodies are primarily made in the mitochondria of the liver, and it's triggered by low insulin levels and high glucagon levels. So once this is in the blood, once these levels are low or high, they trigger adipocytes, which are known as fat cells. And these fat cells, they break into free fatty acids. These free fatty acids they undergo beta oxidation to provide acetyl-CoA molecules, which are needed to make ketone bodies. So acetyl-CoA molecules go through a series of enzymatic reactions, and once it reaches to acetoacetate, it either makes acetone, which we breathe out, or it reduces to beta-hydroxybutyrate, which is then transported along with acetoacetate into the bloodstream. So this is where the package or the mail becomes important, because yes, BHB, beta-hydroxybutyrate, isn't a ketone body, but it's a package of how a ketone body, acetoacetate, unfolds and is used as an energy molecule. So both BHB and acetoacetate are transported into the blood, but BHB is the more stable one, and that's important too. It's stable, it's more stable than acetoacetate, and that's why it's the more abundant ketone body. So in the circulation, in the blood circulation, it gets transported to the heart, the brain, the kidney, the skeletal muscle, wherever energy is most needed. So once it reaches to this target tissue here, it gets reduced, converted back into acetoacetate. And then what distinguish from ketone body production and ketone body utilization is this enzyme here, the SCOT enzyme, which is the rate limiting enzyme in ketolysis. And the liver doesn't have this enzyme, so that's why it doesn't produce, it produces ketone bodies, but it can't use ketone bodies. So after this, BHB it goes through an enzymatic reaction here, and it converts back to acetyl-CoA, which is shuttled to the TCA cycle and then used for oxidative phosphorylation to make ATP. So yeah, that's important. And then since not only is BHB the more stable and abundant ketone body, it's also a chiral molecule. So first we have the R enantiomer, which is made from ketogenesis. And once this R enantiomer is made, it gets oxidized and has energy and signaling properties. On the other hand, we have the S enantiomer, which is actually an intermediate of fatty acid oxidation. And it also has signaling properties, but it's poorly understood, and it's actually under investigation now, like what these signaling properties are. But for the rest of my talk, I want to focus on the R enantiomer and its signaling and energy properties. So under normal conditions already, we actually make a baseline, we actually make ketone bodies, but at a baseline level around 0.2 millimolar. And when we consume a ketogenic diet or do moderate fasting, it actually increases to 2 to 5 millimolars, and this is considered healthy ketosis. And I'm sure people have heard the dysregulated state and the unhealthy dysregulated state of this ketosis, and which is also known as diabetic ketoacidosis. And this basically gave ketone bodies a bad rap because it was found in urine of type 1 diabetic patients. And this is a state where you don't want to be in, and the levels rise between 20 to 30 millimolars. And it's actually pretty hard to achieve if you don't have type 1 diabetes, even through fasting or consuming a ketogenic diet, unless you're intentionally trying to starve yourself, which is also unhealthy. But it's very unlikely, it's very rare for someone to be a healthy individual to undergo or be in a state of unhealthy ketosis. So why is this important? Well, over the past years, fasting and the ketogenic diet have been in everyone's radar because of their health benefits. So we've all know what a ketogenic diet is, but really, what exactly is it? What does it consist of? Well, it's mainly healthy fats here. Healthy fats only, it's usually 90% of your intake. And and there's a lot of people think that, I can just eat protein, but that's not the case. It's high fat and moderate levels of protein that needs to be consumed to achieve a healthy ketosis state. And this is very different from the Western diet here, which is 77% carbohydrates, and mainly most of it's all carbohydrates and some fats and proteins here. Whereas you don't see carbs here. Very little carbs are consumed when someone eats a ketogenic diet. It's usually maybe 10%, less than 10% of their caloric intake. So this is the ketogenic diet, and it's actually been studied for over a century now as a anticonvulsant treatment. So it treatment for epilepsy here, and these are just some studies that have been done and investigating the ketogenic diet and and the protective effects in epilepsy, especially in childhood epilepsy. But these mechanisms are still under investigation. We don't know why this is such an effective treatment. And it's no rufinamide that we learned on Monday, but it's been an effective treatment for some people. So that's where the ketogenic diet has started from. And so what's known so far, if the mechanisms are still unknown, what do we know about the roles of ketone bodies so far? Well, there's actually a few things. Over the past few years, there have been consistent updates of ketone bodies and their roles, mainly from my PI here. He's been kind of one of the forefront of investigating beta-hydroxybutyrate, the more stable and abundant ketone body, as a signaling metabolite. And there are other groups here who also explain the multidimensional roles of ketone bodies. And it's really exciting. And this has been pretty recent, that BHB has been a signaling metabolite, not just an alternative energy fuel. So these are just reviews that explain possible mechanisms by which they might be helpful in aging. And we know that the original signaling metabolite is actually acetyl-CoA. It gets shunt into the TCA cycle, and the TCA cycle is basically like a metabolic hub for other metabolites. And so this is very interesting to see that there are other signaling metabolites just like acetyl-CoA. So it's just been a very exciting field for us. And so like acetyl-CoA, BHB is also multifunctional. And earlier we talked about the RBHB specifically as a cellular energy production hub. So this can be used as an alternative fuel source. The next one is actually BHB partakes in a non-covalent enzyme regulation. And what I mean by that is, like, in the field of epigenetics, which is really huge, we have our DNA, which is negative, and our DNA wraps around these proteins called histones. And these histones, they have tails that can be modified by epigenetic factors. Here, our DNA can also be methylated, but I won't be explaining that today. But these epigenetic factors are influenced by development, environmental chemicals, and one popular one at USC that they're investigating now is pollution, air pollution. How does that affect Alzheimer's, and how does that negatively regulate aging? And then there's obviously drugs and pharmaceuticals, depending what you're trying to achieve here. And there's also aging, which is an important topic, the main topic today, and the type of diets and how ketogenic diet, fasting, caloric restriction influence our gene regulation here. So, and there are downsides to this too, so there are, in the field, there are targets, or people try to study targets that can prevent cancer progression, or even target autoimmune disease, mental disorders, and diabetes, which is pretty prevalent nowadays. So there are actually, for the next few slides, I'm gonna talk about the epigenetic factor acetylation. These are just marks on the histone tails. One type of mark. There's so many post-translational modifications that can induce or repress gene activation here. So the one I want to focus on is acetylation, and there are two important enzymes here. There's the histone acetyltransferase, which is also known as HAT, and the histone deacetylases, which is HDAC. And these are, so HAT is responsible for putting these marks on the histone tail. So they actually induce or initiate gene activation here. So it kind of opens up the DNA and for further regulations of transcriptive activation. And then there's the HDAC, which promotes transcription repression. So these actually remove acetylation marks and make the DNA tightly coiled up against the proteins here. So you can't really do anything with that. And this is important because with age, actually, there's been studies showing that there's actually hypoacetylation. So there's like less marks at these points, and that's alarming because we need to. There are certain pathways, longevity or promote health promoting pathways that need to be turned on to achieve healthy aging. So that's very important here. And then for this talk here, we would focus on the lysine, which is one of the amino acid residues on the histone tails here. This one's mostly studied for post-translational modification on histones. And so since this is so important, are there inhibitors of HDAC already? Yes, there are. The first one that was discovered was butyrate. Butyrate is a short chain fatty acid. And this was actually been shown to inhibit histone deacetylase activity here. So these are just some studies showing that supports this notion. And recently, maybe not recently, but there's been studies showing that BHB, beta-hydroxybutyrate, is also a histone deacetylase inhibitor here. So the study looked at HDAC activity and the concentration of beta-hydroxybutyrate in millimolars. And you can see that certain HDAC require less BHB to prevent the HDAC activity here. So you can see HDAC 1 and 3 requires release, and then 4, 2, but they mainly inhibit class 1 and 2 HDACs. And these are just transcription factors that are involved in longevity pathways here. I won't go too much in detail. But yeah, so it was really exciting news to also find that beta-hydroxybutyrate had HDAC inhibition abilities too. And this is actually a crystal structure of human HDAC8, and there's the zinc binding group here, domain here. So the hypothesis is that the carboxylic or the hydroxyl group are the main groups that are responsible for chelating this zinc right here. So it inhibits the function of this HDAC, which is pretty amazing. And to this day, there are other investigations on what other HDAC inhibitors are there. So it's pretty exciting. The next multifunctional BHB role is covalent protein modification. And this was actually pretty recent too. So we know that acetyl-CoA, the original signaling metabolite, can actually induce epigenetic modification, but BHB itself can also modify these histones here. So we have a lysine residue in the histone, and we see BHBylation occurring on these lysine residues of histones. And you can see here that this is the flux of BHB-CoA enzyme. CoA is very much concentration dependent. So as you increase the concentration of sodium BHB, this was just their study, you can see an increase of this BHB-CoA. So it has to do with the amount of CoA enzymes and the flux to initiate this modification. And this study was actually done in mice and under fasting conditions. So earlier we learned about fasting and ketogenic diet. They both promote a healthy state of ketosis. So this was very interesting. And to this day, this has been the only study looking at BHBylation on histones or even non-histone proteins. And I hope to contribute into this field too in the next year or so. The last two, BHB actually is a feedstock for other metabolites. And what I mean by this is that, so we have ketone bodies, we have glucose to promote glycolysis, and then we have fatty acids here. Ketone bodies here, you can see that the oxidation of ketone bodies actually promote other signaling metabolites here. So we have HMG-CoA, we have acetyl-CoA, we have malonyl-CoA, glutyryl-CoA, succinyl-CoA, propionyl-CoA. This is, so the TCA cycle, as mentioned earlier, this is really like the signaling metabolite hub here. So not only is ketone body itself a signaling metabolite, but it also is a feedstock for other metabolites, endogenous metabolites. The last thing here is, as BHB being a receptor agonism or antagonism, so then they actually, BHB binds to cell surface receptors, and the two main ones we know today are HCAR2 and FFR3, and that has to do with fatty acid synthesis, lipolysis. So these are, it's really interesting to see that BHB are kind of promoting these downstream signaling, this cascade effect in the cell here. So that's one facet of BHB, as it's it binds to receptors on cell surface receptors to promote changes in the cell. It's really interesting. And then on a less global scale, these are just publications showing that ketogenic diet. So the first one, actually, ketogenic diet promotes a reprogramming of the gut and the liver and induces gene expression changes here in mice. So they found that these gene expressions had a circadian rhythm to it. So it's really interesting to see that the ketogenic diet promotes this. Another research that was really interesting was that it, in macrophages here, resident macrophages here are kind of immune system. The acetoacetate actually triggers a response in macrophages to prevent liver fibrosis. And so this was interesting. And then the last study here that I want to share with you all is that a ketogenic diet, but not a glucose supplement diet, actually triggers intestinal stem cell self-renewal. And these were all done either in mice or in culture. But this is also very interesting because this field is still fairly new, and there's been a bunch of studies showing different signaling properties, and yeah, it's just very recent and exciting for us. So here we've learned that the R enantiomer, the RBHB, actually can regulate some aspects of epigenetics. It can prevent certain inflammation responses, and it can also reprogram or trigger a metabolic switch in metabolism. So how are these actually applied to diseases and aging? Well, with age comes with chronic diseases, and these are just some studies showing that the emerging roles of ketogenic diet in cancer treatment. So we know that it's been long time use of epilepsy treatment, but now people are studying how ketogenic diet can be used in cancer treatment. And or there's also this diet called fasting mimicking diet too, which is similar to, not similar to a ketogenic diet, but not very similar to a ketogenic diet, but induces the same responses and same protective gene pathways. And then there's this study here that looked at diabetes and the complications, so treatment of diabetes and diabetic complications with ketogenic diet. So we know that the ketogenic diet is not good for type 1 diabetics because there are already so many ketone bodies made in their bloodstream. But in type 2, it's a different story. Type 2, it's mainly with body mass index, so like weight gain. So actually people who do it now show that there's promising results in like weight loss and more control of their insulin sensitivity and just their glucose levels. So it's very interesting to see. And also here, ketogenic diet has been also studied in the context of cardiovascular risk factors in animals and humans. because there's this preconceived notion that, oh, if I eat a really high fat diet, it must not be good because there's like cholesterol and it's just not good for my health. But studies have shown that it also triggers these protective pathways here with the ketogenic diet. So that's not necessarily true. And to make it more topical and relevant, there's this study here, which is also our collaborator. This group actually showed that a ketogenic diet activates protective gamma delta T cell responses against influenza virus infection. So this was also interesting, and this was actually applied to this review that just came out maybe a few months ago during COVID time from our group, investigating ketone bodies as immunometabolic countermeasures against respiratory viral infections. So just what we know already, how are we going to apply it to COVID-2 viruses and COVID now? So it's really interesting. This is just a molecular model of how beta-hydroxybutyrate, BHB, can prevent virus replication here. So first, it can inhibit glycolysis, which is important because we know that a virus can hijack what the host's metabolism and kind of go replicate a lot based on their energy, their pathway. And another protective thing is that BHB protects against oxidative stress. So with glycolysis, we have a lot of reactive oxygen species production, and that can be bypassed if someone was ingesting beta-hydroxybutyrate or through fasting from ketogenic diet or other means that I will talk about in the next few slides. And there's also BHB can directly inhibit pro-inflammatory activation. So this NLRP3 is an inflammasome. So if this gets activated, it triggers other pro-inflammatory activation here, which is a good and a bad thing because you do want some sort of activation so your body knows that there's an infection, but there's also a fine line that distinguishes between healthy and unhealthy activation, pro-inflammatory activation. And then there's also, BHB also induces anti-inflammatory pathways, which is shown here that I won't dive into, but other than pro-inflammatory pathways, you also want anti-inflammatory pathways to be activated. And then it also prevents repression of protective gene pathways. Here we go back to the Histone deacetylases. So it prevents the repression of protective gene pathways that I've been talking about. And so you definitely want these activated and turned on for protection. And then lastly, it reduces apoptosis, which is cell death, via mitochondrial permeability transition. So it just prevents the permeability of mitochondria. So not too much gets into the mitochondria and just throws everything off. So when that happens, mitochondria is no good anymore, and it's just going to trigger apoptosis with cell death. And then another interesting part is our group actually showed that a ketogenic diet reduces midlife mortality and improves memory in aging mice. This other group, which is our collaborator now, also showed the same thing. So it's quite interesting. In science, you want reproducible results. So this was actually interesting to see that another group was able to reproduce this, even though this was kind of happening the same time. So it's always a good thing that these results are fairly similar, especially if you want to claim something as big as this, as an extend longevity and health span. So it's actually really neat. So that's another context of how ketogenic diet has been used in studying aging and diseases or age-related diseases. And so there's this problem people run into. They say, well, what if I don't like the ketogenic diet? What if I can't even adhere to it more than a day? It's really high in fat, and I can only eat very moderate levels of protein, or like I tried fasting, but I really can't tolerate it as much. I can maybe do like a 16-hour fast, but just for a day. That's fine. I mean, there, as you can see in the stores now, that this market for ketogenic product is expanding, and it's actually expanding at a rapid pace because of this fad, this trend that's going on here. But if you don't like the ketogenic diet and you can't tolerate fasting, there are other ways you can get into ketosis. And the most common ones you see in stores now are mainly just the salt, but there's also the ketone acid here. So this is just a free acid. It has a pKa of around 4.4. It's very acidic. So it's, to me, it's very hard to ingest. Also with the salts here that you can sometimes see in the stores, these are masked by like salts, which is sodium or even like other cations, but it's very salty, and a lot of salt is also not so good. So these were preliminary efforts to kind of kickstart how we can mimic this healthy level of ketosis without consuming a ketogenic diet or fasting. So these were just some of the products or some of the means that have been studied in literature. And then there's also ketone esters, which looks like it's being more favored because it reduces the acidity of the acids and the salts here. And it actually, so a ketone ester is actually a ketone body attached to a ketone body precursor. So here you're making twice as much because you have the ketone body already upon ingestion, and then your body recognizes this precursor and it's going to trigger ketone body production in the liver, which I will be explaining in the next few slides. But it's basically, that's basically it. You attach a ketone ester or a precursor to a ketone body, and then you have ketone esters. And then, yeah, so these are just some examples of ketone esters here that are studied already in the field. But the main two ketone esters widely known are the one is the 1,3-butane-diol, and then there's the medium chain triglycerides that you might see in the stores here. So the 1,3-butane-diol here is it's metabolized through the alcohol metabolism, and it produces the ketone body here, and then it gets broken down to acetoacetate and produce energy or other signaling functions. And then the medium chain triglyceride actually, it metabolizes through fatty acid oxidation and acetyl-CoA, and it's not insulin sensitive, so it will still trigger production, whether it doesn't matter based on your insulin levels, basically. So it produces BHB, and also the same means it produces the acetyl-CoA molecule and energy. And then, yeah, so here you have the ketone ester on the left. So how is this different? How is the metabolism different from like a ketogenic diet or fasting to ingesting exogenous ketone bodies? Well, so we're familiar with the traditional way where we, upon consuming a ketogenic diet, we kickstart our body to break down fat cells right here. So earlier I explained that free fatty acids undergo beta oxidation, and that triggers ketone body production in the liver. On the other hand, if you ingest ketone esters to achieve the same thing, we have here, this is just an example, but pretend this is a ketone ester here. The triangle is a ketone body precursor, and the gold or the orange is a short chain fatty acid. So here, upon ingestion, this ketone ester is actually broken up by esterases, which are found in the GI tract or in like blood circulation. So that gets broken down. And so you have the ketone body precursor, which goes through aldehyde dehydrogenase, and then you have the short chain fatty acid here that undergoes beta oxidation to make ketone bodies. So you're making essentially twice as much as when you have a ketone body precursor and a ketone body. And these are just some tools now that are being studied by different PIs at different universities here. So the traditional is the ketogenic diet, and it raises blood levels to 0.5 to 2 millimolar at most. And it's been studied in the context of obesity, type 2 diabetes, Alzheimer's disease, traumatic brain injury. There's also MCT oils that I just went over. This also raises your blood ketone levels to 0.5 to 1 millimolar. And this typically lasts from 2 to 4 hours here. And the context it's being studied is Alzheimer's disease. There's also the ketone salts I talked about here, raises around the same levels of MCT oils. It lingers around in the blood, maybe 1 to 2 hours, and it's studied in heart failure, in the context of heart failure, and how to boost athletic training, endurance. And then there's the most promising one to date is the ketone ester here. It raises the blood levels the highest amongst all of these, just explained, and it lingers in the blood circulation for 1 to 2 hours. And it's studied by our group too in the context of Alzheimer's disease, athletic training, and TBI. So these are just examples and what's undergoing now and how these can be translated from mice or rodents to humans. And there's this study that came out this year looking at the metabolism of exogenous D, which is the R enantiomer, beta-hydroxybutyrate, an energy substrate avidly consumed by heart and kidney. So here you can see that they looked at MCT oils, a ketone body with a mix, so like a mix of D and L enantiomers, and then there's also the D enantiomer that looked at. And here you can see that the R enantiomer is it peaks at it produces the highest plasma ketone levels, and it peaks at 1 hour, similar to the MCT oils and the racemic mixture here. And then for when they looked at plasma BHB levels, they saw, of course, as expected, that the D form would produce the highest plasma D ketone body, followed by the MCT and then the racemic mixture. I'm not totally sure if it's racemic, but it has both enantiomers. And then they looked at plasma acetoacetate. So how much of this BHB ketone body is being converted to acetoacetate. So it looked like there's a similar trend with the DBHB versus the MCT and the mixture of D and L. And then as expected here, in the mixture of both D and L enantiomers, you can see that the plasma L levels were highest in the mixture and not so high or at all with D, which was expected. And here they just looked at incremental area under the curve and quantified it, and you can see that the RBHB was actually the most promising exogenous ketone body form for ingestion and metabolism. And here, same study, they did a whole body ketone body PET scanning here, and they actually infused radioactive acetoacetate after 2 ingestions of the RBHB form here. So you can see that it was where this acetoacetate was taken up or uptaken, and it was mainly in the heart and the kidney, as you can see on this graph. So they looked at % dosage per gram tissue during this time, and you can see compared to everything else, the liver, the blood pool, the gray matter, the heart and the kidney were avidly consumed the most. most. The uptake was the most in heart and kidney. And then, so that brings me to, so like after explaining all this, like what's going on in this field and how there's a push to study the ketogenic diet, the benefits of beta-hydroxybutyrate, where does my project come in? Like, how do I study ketone bodies? Well, with age, we know that glucose utilization decreases with age. So we become less receptive with, we were less receptive to glucose, and like, especially in our brain, since it's like a high demand in energy, we need something to compensate for this. So earlier I talked about ketone bodies as alternative fuel. So when glucose is scarce, we can use ketone bodies, which lucky for us, we have a backup plan. And there's also other metabolites like pyruvate and lactate too that can be used as immediate energy, but for a more sustainable long-term use, ketone bodies would be an option, the option. And then, so my study focuses on how ketone bodies are a compensatory effect for glucose hypometabolism, especially in the brain as we age. And with aging, actually, glucose utilization decreases more in the frontal lobe here. The frontal lobe is mainly in charge of motor control, concentration, planning, problem solving, speech, and smell. But with Alzheimer's, it's actually where the hippocampus is, and that hippocampus is located in the temporal lobe. So very different sections of the brain that experience glucose hypometabolism. So it's very interesting to see different areas aging different areas in the brain aging differently. So there's a study here that there's the same group who did the exogenous BHB. They looked at the brain, and they looked at regular control brains, mild cognitive impairment brains, and Alzheimer's disease. And they looked at the glucose uptake and the acetoacetate uptake, which is the ketone body. In control, you can see that, yeah, so glucose uptake isn't quite impaired, but also I think age was also a factor. I think these were healthy adults. And with mild cognitive impairment, you can see here that, yes, there's still some glucose uptake, but not as much as the control that we see up here. And then with Alzheimer's disease, you don't see much of glucose uptake at all. But if you look on the other panel here, there's an increased uptake of acetoacetate, which is interesting. So here you can see that glucose, brain can use glucose as energy when glucose isn't responsive or can be uptaken across the blood-brain barrier. So this is interesting to see. And then also with mild cognitive impairment, how there's some slight ketone body uptake too, but not so much in the normal aging brain, because the normal aging brain doesn't really need it. It actually prefers glucose more over ketone bodies for a more sustainable long-term use if it's available. So my question and my research question is, so if glucose metabolism changes with age in the brain and probably other organs too, does ketone body metabolism change? Like it's curious, like if one changes, how about the other? We know there's maybe a shift when a compensatory effect is needed. But my main question is, in a normal aging context, does ketone body metabolism change with age? And if so, how does it change? So I'm currently studying mice in different ages, and female and males, to see if there is a sex difference. So that's basically my main research question. And then going back to BHB, it's a chiral molecule. So does BHB, with age, does BHB impose more on its energy functionalities or its signaling? That's an interesting question here, because no one knows. We only know that BHB is capable of being an energy molecule, and then it also has signaling functions here. And we're also looking into SBHB too, and what it can be doing So it'd be interesting to see if SBHB is actually the more predominant ketone body with age, but it's very unlikely because it's slower. It's metabolized slower, and its excretion. It's usually excreted out in the urine. So yeah, that's where I come in. And then yeah, basically, what I just said, there's like a tug of war between energy and signaling. So how does this change with age? Is it more of a signaling metabolite, or is it more of an energy fuel? So we don't know. And then there are some gaps that my studies or this study may help answer. So how does age influence interventions like the ketogenic diet? So studying different ages, like if I were to consume a ketogenic diet and someone else who's older than me consumes a ketogenic diet, are these effects the same? We don't know. Also, are we able to switch from using glucose to ketone body BHB as an energy fuel as we age? So I talked about this too, and like the aging brain and how it can be a fuel. Also, rather than being an alternative fuel, could it only exhibit signaling functions with age, which I just talked about? And how does tissue-specific metabolism of ketone bodies change with age? We know that the heart, the kidney, the skeletal muscle, the brain, they all can use ketone bodies. But is that preferred over, are ketone bodies preferred with age in these tissues? We don't know. And what are the metabolic fates of ketone bodies with age in tissue or just in general? We also don't know. We know that it gets consumed, oxidized into acetyl-CoA, but once it gets traveled or shunt into the Krebs cycle, we don't know the fates of these. What does it produce after that? So that's also an interesting area of research. And then also there are drawbacks. So obviously there are clinical trials going on now looking at ketone salts, acids, ketone esters, how they can be used in age-related diseases, but a lot of things to be cautious about. So the efficacious dose and duration of ketone bodies or ketogenic diet, how much is enough? Because we can't just give someone a maximum dosage. That's just like a drug. The dose matters. And there are also other additional candidate interventions too that also plays well with downstream mechanisms of specific context. So how does this interfere with other pathways, other specific mechanisms? And especially if you want, like any other drug, how does one drug affect another drug? So how does the diet interact with other factors that might be going on in the body? So those are some drawbacks. And then we all know that aging is natural. But what I have to say today about ketone bodies, we also know that aging is plastic. So we can, in the context of ketone bodies, we can manipulate certain pathways and have it modulate aging in our favor and to promote more healthier lifespan, or even achieve longevity. So even though aging is natural, I just wanted to get across that it's also plastic, that we can do certain things to prevent these hallmarks here that go on with aging. And even though there's a lot more to be discovered and understood and explained, I'm pretty optimistic about my further understanding of ketone bodies and how they can be used in the context of aging. And with that, I want to thank everyone's attention. This is just some aging humor that my colleague and I came up with. These are just some specific model organisms that we use in the lab. So we have the C. elegans, which is the worm, and it's aging with elegans, and then time flies, and then with the mouse, it's I got a tail for you. And so if anyone else has any other questions, further questions, they can email me here. I've listed my email here. But other than that, I want to thank everyone. So yeah, and I think there were some chat questions. But yeah, thank you.
It was fun. Lots of fun.
Anyone have any questions?
Yeah, I actually do have before we start, I was actually asking Dr. Kaluri as I asked if this talk is related to the keto diet.
Yeah.
Right?
Yeah.
So we are right. This is about that. But the focus is more on aging in less than, lose weight, lose fat.
How to trigger those pathways, protective pathways downstream. It's not all about weight loss, but it's also important. Type 2 diabetes is also a problem in society too. So
yeah,
Brenda, Brenda, I just have one question.
Yes.
Are there any specific proteins or any specific foods with say a certain type of proteins that you recommend, or proteins in general are okay?
I know there's a big people study to branch change amino acids, which are not good. They actually don't extend lifespan or health span. So if you can avoid branch change amino acids, but I don't have a particular answer for you. But I know methionine is not good for you. I don't know what good ones would be. Maybe, yeah, I'm not too sure, Dr. Fleury. Yeah, I'm not sure.
Do you have any questions, Gerald?
No, I'm good.
Yeah.
I'm good. I just really enjoyed it.
Me too.
Very good. Excellent presentation, actually. Very good.
Yep.
Yeah, very, very good. Well, I have a question, but you mentioned about this improve the memory of the mouse, right? In the slide. How do you measure the memory of the mice? How do you measure quantize?
Behavioral testing. So we have a mice, and so like one good example is like we have these Lego blocks. We condition them to recognize a specific Lego piece or whatever toy. So we condition them. We expose them to this piece, and I have them get familiar with this, and then take them back out of the arena, and then put them back again, and put two toys there, and see where do they go first. Something they recognize, or some people, one indication of aging of the brain or Alzheimer's is that they're not really interested in their curiosity isn't there. And mice they're pretty curious. So if you see that lack of motivation of like they don't really care, they're just roaming the premises and they're not really sniffing the objects or anything, then that's a typical sign of the aging brain and some Alzheimer's. But there are different studies that behavioral testing that also that are also used to assess cognitive functioning.
Yeah, I think I think you mentioned more the anti-aging on the skin, right? More on the skin, and also I think for the keto diet, mostly people are more focused on the fat, the body fat. Do you think this longevity might be related to actually the loss of fat, or some other specific factor, make it prolong the lifetime?
Obviously, yeah. So I think for me, my best advice is to not focus on just one mechanism. So like you can't be spending your whole life just burning fat. That's also not healthy. You also need fat, especially brown fat. Not so much of like the adipose fat or the white adipose tissue, but it's also being metabolically flexible too. So being able to metabolize carbs, produce glucose, and then also metabolize fat. So I think if you look at it in that standpoint, to be metabolically flexible, I think that can also go a long way with just being healthier aging, living longer. So just being flexible with it, and then you've heard people say like in moderation. It's actually true. You don't want to be on the extreme of both ends. So I think moderation is also key.
Yeah, and I have heard from those keto people, fitness people, they were saying, if you do too much fasting too long, and I think the products of the ketogenic is actually toxic. Is that true? If it's too long, it might have something.
So if you're doing too long, like I said earlier, if you're intentionally starving, you're gonna be producing a lot of ketone bodies, and these are acidic, right? So you're gonna put your body in ketoacidosis, which is the same phenomenon as type 1 diabetic people, and that's toxic because you have no control over that, and your body is just producing a bunch of ketone body, and that has a lot of negative effects too. It just kind of shuts everything down, and your insulin, your glucose, everything just throws everything off. So that's also to avoid. So yeah, you can't overdo it.
And where's the name keto came from? Does it any special meaning?
No, it's actually ketone body actually. So I guess based on chemists, they would just say acetone would be the real ketone body, and it's true. When you are fasting, when you're consuming a ketogenic diet, your breath actually it's a little sweet, and that's because of the acetone you're breathing out. So that's like the main ketone body here. But then ketone bodies actually expanded because when you're making ketone bodies, it's from acetoacetate, and that's also, it has a ketone body group. But the main reason, or the main name, the ketone, is the acetone group, like the acetone and like the ketone body group and the acetone. So,
Oh, okay. So you're saying the keto is actually acetone. Is derived. There's no some special meaning.
It's not. It wasn't some guy's name or anything.
I think the acetone is the end product, but there could be some intermediate stages where it passes through certain chemicals which contain the keto function, but not actually exactly ketones. Some keto function containing complex molecules.
Yes, that's also possible. Yeah, so acetone is usually one of the byproducts, and in humans we usually just breathe it out. So, but it was interesting. Like the main focus or talk was the beta-hydroxybutyrate, which isn't really a ketone body, but it has all these signaling functions. It can be used as energy. So that was the main point.
Yeah. Okay. Yeah. So when people are talking about this lose weight, that issue, they also kind of refer to, there is a case. I think there's the princess. I think the wife of the British UK prince. I think she was known to use the protein. It's some kind of lose weight method. So she remained very slim after giving birth to 3 kids. And I think they were saying they the first week they gave her the nutritious, give it all protein, and kind of a different way of doing things. I'm just wondering, those kind of things also might be related to the keto behind it.
Yeah, maybe. I've never heard of that, but it's interesting that you mentioned, because in development, when newborns, they actually have they also produce a lot of ketone bodies because of the developing brain. So the brain needs all the energy it can take. So then a lot of newborns actually have higher levels of ketone bodies. And then when you're developing as an adult and stuff, it's not so much. And I don't know if this is because glycolytic activity is at its peak, or maybe because our diet has been transitioned from a healthy to a less healthy diet, from just a lot of carbs. But there are certain development stages where you'll see higher levels past baseline, of course, of ketone bodies. So it's actually really interesting. But yeah, I've never heard of that protein or the diet something, but I should probably look into it.
I'll find if I can send it to you.
Okay, sure, thank you.
Yeah, and these days this keto is very hot, and in the health food section in some store they even have the thing you talk about. You can eat, some kind of food directly,
Yeah, it's difficult because when something is expanding that large and that fast, I would be cautious because it's always a money maker. So most of the products you find are not really what they say they are. Some of them might have sugar, and you don't want that either. Some use other sugars like monk fruit sugar, stevia to mask everything, but it's kind of hard to gauge that.
It is hard, and actually there are different ways. Some people saying during the fasting, even stevia, alcohol, sugar, alcohol, they will still increase the insulin level. But some people say that's okay, but some people say even coffee would not be okay. But some people say coffee is fine during fasting. So it's all mixing around.
That's where it gets complex because every individual has different genetic background. It depends. It really depends how your epigenetic landscape is. In development, there were probably some stressors that activated your gene, like regulated your gene some way other than like you're expected or used to. So, we all come from a different background, and some things work for them. Like for fasting, it works great. For some people, ketogenic diet works great. Some it doesn't. Some people actually gain weight. So it's hard to just get it in a general perspective.
Yeah, it's very interesting chemistry, so important and fun, the improving the quality of life and also extending the life. Of course, as you said, those people, the people in fitness or the diabetes, they took in a different track. But it's very exciting to know that this actually, improving the expand of life as well and the quality of life.
Thank you very much, Brenda. Thank you.
Thank you. Yeah.
Hope you enjoyed it.
Very much. Yeah.
All right. Well, enjoy your weekend. Enjoy your Friday. Stay healthy. Stay sane. I think that's the most important thing at this point. Staying sane. And just do what you love when you can.
Thank you.